Good afternoon, everyone. First, I would like to thank all the organizer committee for the kind invitation to give a brief talk in this spectacular meeting, and I wish to see all of you face to face, inshallah, next year. So at the end of this presentation, you should learn the following terminology: HLA antibodies, what's the difference between calculated CPRA, current CPRA, and cumulative CPRA, HLA antigen or HLA typing, and finally, I will wrap up the presentation by talking about the virtual crossmatch. So what HLA stand for? From its name, H stand for human, L stand for leukocyte, because all the initial experiment was done on white blood cells. Now we know that HLA is actually expressed on all nucleated cells. And finally, antigen, because it can elicit an immune response in the form of antibodies. So there are two types of HLA, HLA class 1 and HLA class 2, and both are structurally different. Class 1 is formed of a one big polypeptide chain, as you can see in the left figure, and a small beta-2 microglobulin. Both are encoded by two different genes. We don't do typing for the beta-2 microglobulin since it's the same for all individuals. And we only type for the alpha gene because it's the most polymorphic gene. The most common type for HLA class 1 are HLA-A, HLA-B, and HLA-C. HLA class 2 is a little bit different. It's formed of a two polypeptide chain of equal size that is encoded by two different genes, the alpha and the beta. The most common of the HLA class 2 are HLA-DR, HLA-DP, and HLA-DQ. So basically anything which starts with a D is considered a class 2. As I mentioned before, that HLA is expressed on all nucleated cells, so you would expect that they are also expressed on the kidney cells. So let's see here an example on how or what's the mechanism of antibody-mediated rejection. So what you see in this diagram is a kidney cells expressing HLA. If, for example, a patient got a BK virus and we were urged to reduce the immunosuppression, now we are giving enough room for a donor-specific antibody to form that will bind to the HLA antigen present on the surface. In turn, this will activate the complement cascade complex through the binding of the C1q to the FC portion of the antibody, which leads to recruitment of consequent complement component, leading to the formation of the MAC or the membrane attack complex, causing cell lysis and death. Donor-specific antibody is done in the HIL lab or the Histocompatibility and Immunogenetics lab from the patient's serum. C4D and all signs of kidney damage is seen from the biopsy that is sent to the renal pathologist. There are three types of antibody-mediated rejection, or AMR. The first is hyperacute rejection, which happens between minutes to few days. This is very rare type, as we screen all our patients before transplanting, and we screen for all the possibility of donor-specific antibody, which I'm going to discuss later in my presentation. Acute, which occur weeks to year, and is due to the formation of a de novo or a new donor-specific antibody. Chronic is the most common one, and it's due to the graft or the gradual loss of function of the graft. So we work very hard in the lab to prevent this from happening. And let's see what we do in our lab as a pre-risk assessment for any patient undergoing transplantation. So the first thing we do in the lab for any patient undergoing transplantation is to do an HLA typing. So this is all the steps that most of the HIL lab or the histocompatibility lab follow for HLA typing. First, the samples are received in the lab and then they are accessioned. After accessioning, the samples go to the DNA bench for DNA extraction. Depending on the urgency of the sample, it may be typed by low resolution typing using real-time PCR. And this is usually done during on-call or in the presence of a deceased donor. It takes around 90 minutes to be completed, or routine samples are done by high-resolution typing using the sequencer. And this takes almost between 3 to 4 days to be completed. At the end, all the results are reviewed, and then it's go to the computer to be reported by the lab information system. So this is an example of one of the HLA typing reports from our lab. And you can see here is the HLA class 1, which is the HLA-A, HLA-B, and HLA-C. And if you notice here, there are 2 alleles because each patient received 1 haplotype from the mother and 1 haplotype from the father. If you see here, you can see that the A1 or the A0101 is only 1 haplotype, but actually this means that this patient is a homozygous haplotype. So if you see something like this, don't confuse yourself that the lab missed it. It's actually because the patient is a homozygous, means he has the same copy from the maternal and the paternal. In the second part of the report, you can see the HLA class 2, and the HLA class 2, you can see the DR and the DQ and the DP. The second risk assessment that we do in the lab is to do HLA antibody. And from this figure, you can see that the HLA antibody is formed against the HLA antigen found on the cell surface. So usually the antibody is named against the antigen that it's bind. So for example, if the antigen is an HLA-A2, then the antibody will be called HLA-A2 antibody. Again, in the lab, we receive the sample. The sample is accessioned. And then after the sample is accessioned, we put some of the reagent. And then we put the samples into the Luminix machine. And then it takes around half an hour or 45 minutes to be completed. And then the results are reported into Histotrack, which is our lab information system. Here is an example of a report that comes from the machine. We don't share this report with the physician, but I would like to point some important things for the purpose of this presentation. So what you see in the y-axis is the mean fluorescence intensity, or the MFI, which measure the antibody strength. What you see in the x-axis is the HLA antibodies. You can see that the highest bar is at 12,000 MFI, and it represents the B48 antibodies. The cutoff for antibody in our lab is 1,000 MFI, which means anything below 1,000 is considered negative, anything above 1,000 is considered positive. Based on that cutoff, the last antibody that is considered positive will be B76. So in conclusion, what you see highlighted in red are all our positive antibodies, and anything highlighted in the blue box are considered the negative HLA antibodies. So this is just a disclaimer when you use the mean fluorescence intensity or the antibody strength. So for example, if your lab are actually sharing with you this information or giving you the mean fluorescence intensity, you have to use it with certain cautious, because there is a lot of limitation, and MFI cannot be used as a quantitative metrics for antibody amount. First, because there are tech-to-tech variation, and also there is a run-to-run variability. Even you can see different results between different labs. If the labs are using different lot number, it will have also different results. And also for target epitopes, because now we know that antibodies are against epitopes. They are not targeted against the antigen, they are actually targeted against epitopes. So if there is an epitope spread between antigen, then you might get weaker MFI. And again, and the last thing here is the presence of some of the interference substance present in the serum, like which can cause inhibition or decrease the MFI, like the presence of IgM antibody, or the presence of some complement cascades or complement like the C1q. So the most common question that we usually ask, how do we get sensitized to HLA antigen? So the most common thing is transplantation, especially if this is a failed transplantation. The second thing is pregnancy, because now the infant carry half of the HLA comes from the dad. Blood transfusion, and here I'm going to stop for a little bit because since 1990, or at least in Canada, all the red blood cells or the packed red blood cells are leukoreduced, means they remove as much as they can from the white blood cells. So I believe they can guarantee up to 99.9% remove. So there is a small leftover that can result. But here the question is, how many blood units are recommended until you get sensitized? Sometimes we don't see anything. Sometimes we can see at least like 10 units of blood. But the other thing here which I want to point is if the patient is taking platelets, then the story is completely different, because platelets is a big source of HLA antigen, and from 1 unit you can get highly sensitization, even can reach 100 PRA. And I'm going to explain this in the next few slides. The last thing that can cause sensitization is severe infection or even vaccination, and we have seen this with some of the COVID vaccine that I will present, or being presented in the other case. Sometimes the HLA antibodies produced from vaccination are just transient, but in some cases they actually persist. So in the next few slides, I will explain the difference between calculated PRA, current PRA, and cumulative PRA. So I will start with the calculated panel reactive antibody, or CPRA. And CPRA actually predict the percentage of donor in a specific population, and I put 3 lines against specific population that are likely going to be incompatible with a given recipient. So how this is calculated? It's calculated based on the allele frequency of a specific HLA alleles in the population. I don't want it to get so complicated, so I'm going to explain it briefly now. So for example, if someone did their PRA in Asia or Africa or South America, their CPRA will be different than if they did it in North America or in Europe. And the reason, because the allele frequency or the HLA allele frequency is different between population. So for example, if we took HLA-A2 as one of the example, HLA-A2 is one of the most common HLA alleles in the Canadian population, and it accounts for about 40% of the population. So if we have 10 individuals, we would expect at least 4 of the individuals will have the HLA-A2 allele. This means 4 out of these 10 has an HLA antigen onto their cells. Now, if we have a patient who has an HLA-A2 antibody, then they will be incompatible with 40% of this Caucasian population, because their cross-match will be positive. So based on the example that I just mentioned, this patient will have a CPRA, or a calculated CPRA, of 40%, because he will react to 40% of the population. Now let's talk about the cumulative and the current CPRA. So every time a patient has been exposed to a sensitizing event and form a new antibodies, these new antibodies will be cumulatively added to the previous one. And these are HLA antibodies that has been detected over the entire course of the pre-transplant screening. So for example, if a patient got listed in 2010 and then in 2011 they develop antibodies. Then these antibodies are added. Say in 2015 new antibodies are developed. Then this new antibodies got added to the previous one, and so it's and so on and so on and so on. So it's actually cumulative. It doesn't get deleted because antibodies comes from memory B cells, and memory B cells, even if it's down or up, but if this patient got exposed again to the same antigen, it can boost again. So this is why this is important, because when lab performs the virtual cross-match, which I'm going to explain in the next couple slides, they depend on all the cumulative PRA. They don't use only the last PRA, but they actually use the cumulative CPRA. So my advice for new physicians is, if they are going to use, or if their patient is going to use to do antibodies, is to stick to one lab when they do this antibodies. So please don't use different labs, because as I mentioned before, every lab will have a different cutoffs and every lab will have a different ways of calling antibodies. So let's take an example here. So HLA-A2 antibody was detected in a patient in 2002. But in 2023, the patient did not have any antibody in this test. So now the test that was done on 2023, this is the current CPRA, and it is 0% because there is no antibody detected in the last one. But his cumulative PRA will not be changed because he has historical HLA-A2. So now his cumulative CPRA will be 40% because of the historical A2 antibody. And cumulative CPRA is the one that we used for virtual cross-match. And here is an example of a report that you should receive from your lab. The top part is the current serum, which means latest serum that was tested for antibody, and it's 0 PRA. In the bottom of the report, it's the cumulative CPRA, which is 25.48%. And this antibody is accumulating all over the years. And the patient have some unacceptable antigen, as you can see, A3, A66, and B76. So here I'm going to start talking now, or switching gear, and talk about the virtual cross-match. So what is the virtual cross-match? Virtual cross-match is used to predict the result of the physical cross-match. And in order to do a virtual cross-match, you need to have the following: the recipient HLA typing and the recipient HLA antibodies. And again, I remind you that we are using the cumulative HLA antibodies. From the donor, we only need the HLA typing of the donor. So how we do this in our lab? So all wait-listed patients have their HLA typing done in their initial assessment. In addition, in order to be maintained in the wait list, we have to draw a serum samples once every 3 months for HLA antibodies. The HLA typing and the cumulative PRA are now added to our computer system that has the software for the virtual cross-match. Now, if we received a deceased donor typing, or even a live donor typing that wants to donate their kidney anonymous, we do an HLA typing for this donor, and we add the HLA typing to the same software. The computer software will compare the donor typing to all the cumulative antibodies of all the wait-listed patients, and will generate a virtual cross-match report. The report will contain all possible waitlist patient that is predicted to have a negative cross-match. Patient that is predicted to have a positive cross-match will be removed by the software. And here is an example of one of the virtual cross-match report that comes from the computer. On the top part, you can see that the deceased donor typing, and you can see that the ABO of this donor is group A. In the bottom, you will see all the wait-listed patient that is virtual cross-match negative to this donor, and they are all blood group A. All the HLA typing of the HLA typing of the patient will be displayed, and now in the next 4 columns is filled manually when we are comparing the HLA typing of the recipient with the donor. For example, the first patient has an A2 and A11, but the donor has A1 and A2. So they share the A2 antigen, but they differ in the A11 antigen. So they get the score of 1 because there is only one mismatch. And in the second column here, patient is B13 and B35, and the donor is B8 and B39. This means they have two mismatched, so they get the score of 2. So you will see a score of 1, 2, and 0. 0 means there is no mismatches between the donor and the recipient. And this is actually very important when they pick from this list, because the first thing our transplant nephrologists see is the patient that has the less mismatched to the donor. Now let's see how the computer actually do the virtual cross-match. So what you see here is the disease donor HLA typing report. And this is a recipient ABC HLA antibodies. The computer will compare the compatibility of the donor to the recipient by looking at the antibodies in the recipient that can bind to the donor. In this example, the patient has, or the recipient has, CW12 antibody that can react with the CW12 antigen, has DR1 antibodies that can bind to the DR antigen, and lastly has DQ5 antibodies that can bind to the DQ5. So in this case, the patient has a positive virtual cross-match for both class 1 and class 2. Now here is another recipient. I call it recipient 1 to 3, but it's the same donor. But as you can see that this recipient has some antibodies, but none of these antibodies can able to bind to the recipient HLA typing. So in this case, the computer will pick this recipient as a virtual cross-match negative. So the take-home message of this presentation is that we don't do HLA match everyone, but we make sure that they are compatible. We know we need to avoid antibody-mediated rejection, so we pick a recipient who doesn't carry HLA antibodies against the donor HLA antigen. We do our best to make sure the waitlists don't miss any opportunity, but at the same time have a successful transplant. Thank you for your time, and thank you for listening. I will